Great. And then let me reshare. Okay, so I want to welcome you to our monthly CCEH Meet the Expert seminar series. And today we're delighted to hear from Vijay Sankaran, who's at Boston Children's Hospital. Before I introduce Vijay, I want to just remind you of going to the CCEH website, which is located here, cceh.io, and you can find, this is the homepage where you can find up and coming seminars here. You can find opportunities to apply for funding. And I just want to hit that link for you because there's a new call out for funding. These are funding opportunities to use cores that are at the different hematology centers around the country, where you can apply for type A pilot and feasibility monies that are core credits for you to utilize the cores that are at the different core website, different core facilities across the country.
Dr. Klein, I'm Jane Nieberger. I'm the Associate Chair for Academic Affairs in the Department of Cardiology.
Sorry about that. I can see I need to mute people, and I will, let's see. Yeah, okay. Anyway, let me just tell you about these Type B awards, which are up to $75,000, which are P&F awards for new investigators, postdocs who can apply for these fundings. The call and due date for these applications for the Type B awards is August 1st, and I highly encourage you to apply for those. And the Type A awards are available to anybody, and they're you can submit those today. You can submit them anytime. They're on a rolling basis to be reviewed and awarded or given feedback either way. And let me stop sharing for a minute. And then I want to now go ahead, Vijay. I'm going to introduce you. This is Dr. Vijay Sankaran from is a Lotus Family Chair in the Division of Hematology and Oncology at Boston Children's Hospital. He's the Associate Professor of Pediatrics at Harvard Medical School, an attending physician in the Dana-Farber, wow, today, Boston Children's Cancer and Blood Disorder Center. He received his bachelor's and master's degree from the University of Pennsylvania in biochemistry and a master's at the University of Cambridge in biochemistry, then earned an MD, Ph.D. degree from Harvard Medical School and worked in the laboratory of Stuart Orkin. Subsequently did a residency in pediatrics at Boston Children's Hospital and Boston Medical Center, followed by a fellowship in pediatric hematology oncology at Boston Children's Hospital and the Dana-Farber Cancer Institute. Dr. Sankaran has received a number of awards for his work on using human genetics to better understand the process of blood cell production, including most recently the 2018 Gail and Ira Drucker Prize in Children's Health Research and the 2019 Selden Smith Award for Pioneering Research from the American Society of Clinical Investigation. And I want to welcome you today, everybody, and welcome Dr. Sankaran for speaking to us. And we'll go for about 45 minutes. If you have questions, you can populate them in the chat and I will monitor them. I want to ask everybody if they would please mute themselves at this time so that we can have a real clear presentation. And if you have a question, populate it in the chat, and then I'll invite you to unmute yourself, if that's okay. And again, thank you, Vijay, for your presentation today.
Well, thank you so much, Diane, for the opportunity to come here and present. And it's really terrific, even in these times of being virtual, to be able to see so many, at least virtually, friends and colleagues. So what I want to try to do in the next 45 or so minutes is going to tell you a little bit about, in general, our interests. Many of you will be familiar with those, but I hope to then dive into two largely unpublished vignettes to kind of illustrate the types of approaches that we're trying to take and some new areas that we're expanding into. Now, this audience, of course, is very familiar with this aspect of blood cell production or hematopoiesis I'm depicting here. Now, arguably, we know more about this process than any other example of cell differentiation in normal physiology. We understand a great deal from work of a number of people in the audience and throughout throughout the world about how this process is regulated at a molecular level, as well as how this process occurs at a descriptive level. And certainly our understanding of this process continues to evolve, but I would argue that compared to any other tissue, we know more about this process than most others. But it's important to bear in mind that most of our knowledge of this process really comes from work in elegant model organisms, much of which has been supported by the CCEH, including mice and zebrafish, which have taught us a tremendous amount. And yet, as a hematologist, clinically, much of what I see in my practice cannot necessarily be explained with our current understanding of this process. And so we view this as an opportunity to take advantage of naturally occurring human variation and the diversity that occurs across healthy human populations, as well as in disease states, to better understand this process. And we've been trying to take advantage of this, and this really started with work that I had done as a graduate student in Stu Orkin's lab to be able to better understand aspects of fetal hemoglobin regulation. And this has continued to be an area of interest where we've been trying to continue to take advantage of naturally occurring human variation to gain new insights into this process of fetal hemoglobin regulation. As time has gone on, we've of course been interested in trying to use both rare genetic variation as well as other experiments of nature to better understand how lineage commitment occurs normally and how this process might be dysregulated in disease. And this has led us in studies of Diamond-Blackfan anemia and other bone marrow failure disorders. We've also been interested in trying to take advantage of population-based variation, both of common genetic variation and more recently of rare genetic variation, as we've been taking advantage of larger and larger population-based studies to be able to delineate both variation in blood cell phenotypes beyond fetal hemoglobin itself, as well as in terms of understanding the risk factors underlying blood cancers and other clonal disorders. But of course, as we've been conducting these studies, and certainly we've learned a lot, one question that's remained in our mind is how exactly is stem cell self-renewal regulated? And can we gain more insights by taking advantage of naturally occurring human variation? So the first vignette I'm going to tell you about is really focus on the question of how we can use genetic variation from humans to understand the factors that drive blood stem cell self-renewal. And the work I'm going to be telling you about was really led by three talented postdoctoral fellows, Richard Voigt, as well as Fulong Yu in our group, in a close collaboration with Li-Ming Tao and Aviv Regev's group. So one thing that Richard, Li-Ming, and Fulong had noticed was that there was naturally occurring experiments of nature that potentially could teach us more. So over the past few years, a number of groups have described individuals and patients with heterozygous loss of function of this transcription factor MECOM that result in neonatal onset aplastic anemia. These mutations affect both isoforms of MECOM, and this can include the upstream MDS isoform, as well as the downstream EVI1 isoform, and can include deletions or nonsense frameshift mutations, as well as a cluster of missense mutations, all of which occur within the zinc finger domains. These mutations can cause some developmental defects, and this can include radioulnar synostosis, illustrating here, but in almost every patient where this has been described, the patients have neonatal onset thrombocytopenia that typically progresses within the first couple of months of life to frank aplastic anemia, typically requiring bone marrow transplantation for survival of these patients. Now, one of the interesting observations has been that when bone marrow has been evaluated from these patients, they almost completely lack blood-forming stem and progenitor cells, as marked here, for example, by the CD34 positive, CD38 negative compartment. Of course, this limits our ability to study the patients directly, given the absence of blood-forming stem cells by the time of diagnosis. Interestingly, mice with mutations in either the MDS1 isoform or with the EVI1 isoform have fairly normal hematopoiesis in the heterozygous state. So, for example, mice that Fernando Camargo has described with loss of function of one copy of MDS1 have ostensibly normal stem cell compartments. And moreover, EVI1 animals, while they do have stem cell defects in the heterozygous state, have and maintain ostensibly normal hematopoiesis throughout their lives. And so this suggested to us that there was a unique opportunity to study specific dependencies of human hematopoietic stem cell self-renewal by taking advantage of and studying this experiment of nature. So since we could not obtain patient samples, we reasoned that perhaps we could use and take advantage of genome editing tools to manipulate this gene in primary stem and progenitor cells, either from cord blood or from adult bone marrow, and to be able to then perform a variety of in vitro studies as well as in vivo studies. So initially, we performed in vitro cultures of these blood-forming stem cells, and we could use a phenotypic strategy that Guy Savageau's lab has described, where we could take advantage of cells that are marked by CD34 positivity, that are negative for CD45 RA expression, that are positive for CD90 and CD133 expression, as well as that are positive for EPCR and IgGA3 expression. This marks long-term reconstituting hematopoietic stem cells. And what we can see is over time in vitro, we see loss of these stem cells after MECOM editing. Importantly, if we then plate these cells at an early time point after editing for colony forming assays, we see that there's a loss of multipotent colonies with an expansion of unipotential colonies, as I'm showing you here. Now, we could then go on and perform xenotransplantation, in this case into the NBSGW recipient that has both an NSG background, making it immunodeficient, as well as the W41 KIT mutation. And this resulted in a failure of engraftment when we edited the cells, as seen both in the peripheral blood, as well as reduced engraftment in the bone marrow of these animals. We could also see that since this was a mosaic edited animal, that the wild type alleles, while very low initially at the time of transplantation, after 4 months of reconstitution actually became the predominant cell population, as I'm showing you here in the bone marrow. Importantly, if we look at those animals that are engrafted, we see similar levels of lymphoid, myeloid, megakaryocytic, or erythroid engraftment, as I'm showing you here. But we do see a reduced amount of stem and progenitor cells that are engrafting in this compartment, suggesting and showing that this model recapitulates what we're seeing in the human setting. So now that we had established a model of this human phenotype, we wondered if we could use this to better understand the transcriptional regulatory networks that were necessary for human blood stem cell self-renewal. So to do this, we wanted to use single cell technologies, in part because we know that there's considerable heterogeneity within the stem cell compartment, and thus wanted to use these technologies to be able to look at specific subpopulations. By sorting using the same phenotypic markers I had shown earlier, we could get a population of cells that was enriched largely for this hematopoietic stem cell signature that we had defined previously, which, as you can see here in a large atlas of approximately 200,000 or 300,000 cells from cord blood, actually enriches a very rare population of cells, about 0.6% of cells that are enriched for stem cells. And indeed, the population we had obtained was largely enriched for this signature, as I'm showing you here in UMAP space. Importantly, if we look at the transcriptional overlap between those cells that are edited at the control AAVS1 locus, a safe harbor locus, or at the MECOM locus, we could see that there was actually significant overlap, as I'm showing you here, suggesting that there were not major differences in the types of cells that we were examining, nor in the global transcription profile of these cells. Since we had a mosaic situation where some cells would be edited to varying extents, although we know that the overall editing efficiency was approximately 40% in these cells, we decided to also perform an orthogonal single cell approach that was lower throughput, where we could genotype colonies and then simultaneously obtain single cell RNA sequencing data from the very same cells that we've genotyped. And using this strategy on a few hundred cells, what you can see is that MECOM heterozygous cells span the entirety of the transcriptional profiles that we're capturing using this UMAP space, really suggesting that we were doing a good job of capturing heterozygous cells. And moreover, we could see that AAVS1 colonies that were edited using a similar technology show similar profiles across this UMAP space. Now, while the global transcriptional profiles were similar, we wanted to tease apart to what extent might there be transcriptional differences. And as you could expect, there were not major transcriptional differences comparing these enriched long-term stem cell populations between the MECOM edited cells and the AAVS1 edited cells. But as you could see previously, there was clear functional differences with a failure to engraft, as well as a reduction in total cells over time. Importantly, we could see that these functional differences were really driven by subtle changes in a network of genes. There was approximately 300 downregulated genes and a similar number of upregulated genes. And while each gene was individually changed to a small extent, there was significant changes. Now I'm showing you the expression of just a handful of genes here. And as you can see, there's many canonical regulators of stem cell self-renewal, such as HOXA9, HOXA7, MLLT3, MSI2, and other factors that were subtly but significantly downregulated. Importantly, we also see a group of genes that are known to be important for differentiation of stem cells out of the stem cell compartment that were actually subtly upregulated upon these perturbations. If we then globally map the group of 300 genes that were either downregulated, what we see is that these genes showed a significant overlap and largely an enrichment of expression in the stem cell and multipotential progenitor compartment, similar to MECOM expression itself. While the group of genes that were upregulated upon MECOM editing, these so-called MECOM up genes as they'll refer to them, were largely upregulated as stem cells and early progenitors exit the quiescent stem cell compartment and undergo differentiation toward the myeloid or lymphoid lineages I'm showing you here. Now, we then wanted to examine to what extent we could actually rescue this phenotype. So we performed the editing as I'd shown you before, either at the AAVS1 locus or at the MECOM locus, and then introduced using lentiviruses, either MECOM or a GFP. We could also introduce other isoforms of MECOM as well to test this. When we edited these cells at the MECOM locus, we could actually get a reduction of MECOM expression. This was at an earlier time point, but we could see an increase in expression using reintroduction of MECOM containing virus that I'm showing you here. And importantly, while we see a reduction in the percentage of long-term stem cells after a few days, we see that this can be rescued by reintroducing MECOM. Importantly, we not only see a rescue, but we can actually expand and maintain these long-term stem cells over time in culture over several days, as I'm showing you here, through the reintroduction of MECOM in this setting. Now, we then looked at global gene expression by sorting the phenotypic long-term stem cells. And importantly, in bulk, we see the same set of genes that we had seen in the single cell data that were downregulated upon MECOM editing, that were once again downregulated in this group of cells. Importantly, though, upon rescue, if we perform RNA-seq in the phenotypic long-term stem cell compartment, we can see a re-upregulation of the large majority of these genes that were actually previously downregulated upon MECOM editing, suggesting that we are not only getting phenotypic rescue upon reintroduction of MECOM, but are actually rescuing the molecular phenotype that's present. We then wanted to try to define potential cis-regulatory elements that were actually impacted by this MECOM-dependent gene regulatory network. We could do this using a number of integrative genomic approaches, both by mapping RNA attack-seq correlations, using promoter capture high-C data to be able to map direct target interactions, as well as define attack-attack correlations using the Cicero platform that Jay Shender's lab has developed. And we could integrate these to actually be able to score different cis-regulatory elements and their potential target genes. And I won't go into this for the sake of time, although I'm happy to talk more about this method later. We could then enrich for those cis-regulatory elements that were specifically accessible in hematopoietic stem cells to attempt to define cis-regulatory elements that were regulating the downregulated or upregulated genes upon MECOM editing. Importantly, when we perform this analysis and examine motifs using de novo motif discovery in the cis-regulatory elements that we defined, we see a profound enrichment for the ETS motif, as I'm showing you here, along with a variety of other factors that I'll describe in just a minute. Importantly, the ETS motif has and shows significant similarity with a known EVI1 or MECOM binding motif defined from ChIP-seq performed in acute myeloid leukemia cell lines previously, really suggesting that many of these sites might be directly bound by MECOM itself. Moreover, we could see robust footprinting in ATAC-seq data that we had obtained, really showing that MECOM or another ETS domain containing transcription factor is likely bound to these cis-regulatory elements. We could also perform footprinting for a variety of other factors that we pulled out motifs for, including RUNX, June, GATA motifs. And importantly, we see a great extent of overlap, particularly between the ETS motifs and the RUNX motifs, as I'm showing you here. Moreover, if we look at ChIP-seq data from bulk stem and progenitor cell populations, we can see that there is significant overlap of other ETS motif containing transcription factors such as FLY1, RUNX1, GATA2, and that these often co-occupy and actually co-occur at many of these cis-regulatory motifs, as I'm showing you in this Venn diagram. Now, we also notice, though, interestingly, not just cooperativity between these different transcription factors, many of which have been shown previously to be important in stem cell self-renewal, but we also pulled out the CTCF motif. And this was of particular interest given recent work from John Dick, Matt Lupien, and other colleagues, where they had shown that in the exit from quiescence of these long-term hematopoietic stem cells, there's actually activation of CTCF and reorganization of the genome that's necessary to enable stem cells to undergo differentiation and to undergo activation, as I'm showing you here. Blockade of CTCF could actually prevent the exit from quiescence and preserve the cells in the quiescent compartment. So we wanted to understand to what extent MECOM might be interfering with the CTCF-dependent activity that's necessary for exit from quiescence. To do this, what we could examine is CTCF binding in either stem or progenitor cells or in erythroid or other differentiated cell types using ChIP-seq. Importantly, what we can see is that CTCF actually is bound to these MECOM-regulated cis-regulatory elements, as I'm showing you here, that are CTCF bound, almost equivalently, and if anything, even better, as cells undergo differentiation. This really suggests that this CTCF binding does not occur in the stem cells themselves, but actually occurs as cells undergo further differentiation. Consistent with this, if we use loci data, either from sorted phenotypic long-term stem cells or from short-term stem cells, if we look at local looping interactions, we can see that there is similar looping either in the long-term or short-term stem cell compartment. In contrast, if we either look at the MECOM down-regulated genes upon editing or the MECOM up-regulated genes upon editing, what we can see is all of these genes show very little looping as compared to the background in long-term stem cells. But these genes actually become much more tightly looped, and there is an increase in interactions as the cells undergo differentiation towards the short-term stem cell compartment, as I'm showing you here. This suggests that actually MECOM is constraining the ability of CTCF-dependent genome reorganization to actually mediate the genomic reorganization that's necessary as stem cells undergo differentiation. I haven't shown it here, but in fact, we've now actually demonstrated that at least in part, CTCF is necessary for this MECOM-dependent stem cell self-renewal activity. And sorry, Diane, was there a question?
Yeah, there's a question in the chat from Dean Tanton that asks, could you walk us through the panels on the right one more time?
Oh, absolutely. I'm sorry I went through this rather quickly. What I'm showing you here actually is data that we have obtained from loci, which is an approach to really look at chromatin loops that occur in a limited number of cells. And what we can see here is at different resolutions. This is looking at 100 kb from the loop anchor, and this is at the loop anchor itself, which is centered here. We're just looking at different distances. And really what we're trying to say is there enrichment? In this case, I'm quantifying enrichment really from 25 kb, which is, approximately partway down here, but we see similar results at different resolutions. We're really looking at the degree of looping. So red indicates more looping, blue indicates lack of loops at different resolutions. And what we're really looking at is comparing to an anchor, what is the degree of genomic looping and interactions with other regions of the chromatin. And as you can see here, on average, there's actually an enrichment at 25 kb in both the long-term and short-term stem cells across all genes. And this is looking at all loops that are present. Really no major change between these compartments. But in the MECOM down-regulated genes, we do see differences between these compartments. Hopefully that.
Yeah, thank you. Dean, does that cover your question? Okay, thanks, he says.
So finally, in discussing this vignette, I've discussed a lot about the cis regulatory network mediated by MECOM that's necessary for stem cell self-renewal. But we also know, on the other hand, that about 10% of acute myeloid leukemia cases actually upregulate MECOM. Many of these cases co-opt MECOM expression by juxtaposing other enhancers, such as this GATA2 enhancer, to then drive expression of MECOM or EVI1, as I'm showing you here. So we wanted to understand to what extent would the gene regulatory network that we had uncovered from studies of normal human hematopoietic stem cells be co-opted in leukemia. And what we could see was that actually, as compared to MECOM itself, this gene regulatory network, for example, the MECOM down genes, if we look at them, are actually, if they're enriched in particular AML cases that I'm showing you here, for example, data from the TCGA, they actually show a much worse prognosis in acute myeloid leukemia as compared to those acute myeloid leukemia cases where this gene network is unchanged or depleted. And in fact, this shows a much more significant impact than MECOM expression itself, really suggesting that this MECOM regulated gene network may be many times co-opted, whereas MECOM expression itself may not be co-opted. I will say, although I have not shown here, that we've actually been able to replicate these results across other AML cohorts, including the BEAT AML cohort and the Target AML cohort, which is a pediatric AML cohort. Importantly, looking at the TCGA, if we simply look at differential gene expression between those individuals who survive for greater than 2 years versus those who do not, we see that actually these MECOM downregulated genes are once again enriched, suggesting that there is indeed an enrichment for the same gene regulatory network that I've shown. Moreover, we could then go on and perform editing in one of these MECOM upregulated AML cell lines, the MUTC3 cells, and we could downregulate MECOM. Normally, these MUTC3 cells exist in a variety of compartments, including cells that are marked by CD34 positive expression, as well as more differentiated or mature cells that are marked by CD14 expression. And by MECOM editing, we can actually deplete cells from the progenitor compartment and actually completely have them occupy these mature progenitor compartments, or these mature compartments. Moreover, if we perform this editing, we can once again see that the same downregulated gene networks that we have pulled out from human hematopoietic stem cells are actually downregulated once again in these AML cells, really suggesting that overexpression of MECOM is co-opting the very same networks that are seen in normal stem cells. And moreover, we can see that similarly, the upregulated genes are once again upregulated upon MECOM editing of these leukemia cells. So in summarizing this vignette, what I've shown you is by modeling rare cases of this neonatal form of aplastic anemia due to MECOM haploinsufficiency, we can gain insights into fundamental transcriptional circuits that are necessary for stem cell self-renewal. I've shown you that MECOM cooperates with a number of other key master transcription factors to enable appropriate gene expression. I've also shown you that MECOM constrains CTCF-dependent looping that's necessary as stem cells undergo differentiation and exit from quiescence. Finally, I've shown you how the same transcriptional circuits are co-opted in acute myeloid leukemias that are actually upregulate MECOM or that harbor MECOM mutations. I'm happy to, if there are any questions about this vignette, kind of take any more additional questions that might be there.
Don't see any right now, but if anybody wants to unmute themselves and ask a question, please feel free.
Great. So, of course, I've described one particular vignette of how we can take advantage of human variation to better understand fundamental biology that can teach us more about stem cell self-renewal, as well as how these pathways might be co-opted in leukemia. But as we've been studying human phenotypes, we've of course faced limitations. We can certainly study blood cell counts or other blood cell traits that are readily measured in peripheral blood, and we can assess the bone marrow of patients. But unfortunately, we're limited in asking fundamental questions about blood cell production. Specifically, for any stem or progenitor cell, we cannot assess to what extent might that stem or progenitor cell undergo differentiation. Does it occur in an equal and balanced manner with all stem cells contributing equally to the mature blood cell or mature tissue compartments? Or does this occur in a biased manner with one particular stem cell dominating the picture and giving rise to most all of the mature cells that are present? And ideally, we would love to be able to address this in humans and in many of our patients. Now, in model organisms, the field of being able to barcode cells has advanced considerably. This really started in our field of hematopoiesis with early work on single cell transplantation. It's evolved with the use of fluorescent markers and other reporter genes. And more recently, has evolved to the point where we can actually be able to tag every single cell during divisions with evolving barcodes, such as have been described by Jonathan Weissman, Jay Shendor, and others. But of course, this has left us limited in our studies of humans. Certainly, we can take advantage of naturally occurring nuclear mutations, but these occur very rarely. So for instance, work from Mike Stratton's group has shown the opportunity to take advantage of mosaic mutations that occur during development to be able to examine cells that are more closely related to one another during development as compared to other cells that do not contain these mosaic mutations. And work from Chris Walsh's lab has shown the advantage of taking advantage of rare insertions and line element changes, as well as mutations that occur in subsets of mature neurons, for instance, to be able to tease apart closer relationships from those more distant. Unfortunately, while these approaches are certainly advantageous and can give us insights in humans, they're somewhat limited. Every single nucleotide variant occurs pretty rarely in the nuclear genome, typically once or every 10 to the 8th bases per cell division. Certainly, copy number variants and microsatellites occur more frequently, but these can be difficult to genotype. Moreover, there can be issues in terms of sensitivity with a high dropout rate of a single allele in a single cell. And unfortunately, even with improvements in scalability, sequencing a single genome is typically costly, with it costing hundreds of dollars per single cell, if not thousands of dollars. And unfortunately, there's no information about cell identity or cell state that's obtained at the same time. So we reasoned, could we actually overcome some of these limitations and identify an alternative approach? And the work I'll be describing was led by Leif Ludwig, a former postdoc in our lab, who's now has his own independent group in Berlin, as well as Caleb LaRue, a graduate student in our group who's now at Stanford, and Jacob Eulersch, in another close collaboration we've had with Aviv Vergev. And what we realized is that in contrast to the nuclear genome, we could take advantage of mitochondrial DNA mutations to be able to perform lineage tracing. Now, the mitochondrial genome is very convenient since it's very small in size, being 16.6 kb and encoding 37 genes, almost the entirety of which is transcribed. Importantly, compared to the nuclear genome, there's hundreds, if not thousands, of copies per single cell of the mitochondrial genome. And this facilitates mutation detection above certain thresholds and enables us to avoid dropout or other issues that can occur with the nuclear genome. Importantly, there's a much higher mutation rate given the lack of repair systems in the mitochondrial genome as compared to the nuclear genome. And this facilitates acquisition of mutations with every cell division. And importantly, there's mutations that can occur in a subset of mitochondrial genomes, the so-called heteroplasmic mutations, or there could be mutations that occupy every mitochondrial genome within a cell, so-called homoplasmic mutations. Now, such mutations have actually been used in the past for lineage tracing, but this has occurred in the setting of specific mutations. For instance, loss of function mutations in cytochrome C oxidase that occur within intestinal stem cells are then propagated to all of the progeny of that stem cell within a specific intestinal crypt, and therefore result in negative staining for cytochrome C oxidase. And when these investigators then pick off these colonies and sequence them, they can see the same exact loss of function mutations in all of the stem and progenitor cells, as well as all of the progeny. So this shows the value of individual mutations for lineage tracing, but we wanted to ask whether we could use this approach in an unbiased manner without knowing anything a priori about the mitochondrial mutations present and use it as a lineage tracing tool. Importantly, we could take advantage of commonly used single-cell genomic approaches, including single-cell attack sequencing, at least earlier iterations that allowed us to capture the mitochondrial genome robustly given excellent transposis accessibility of the mitochondrial genome, as well as full-length RNA sequencing, as shown here, for example, with SMART-seq2-based approaches, where we can get excellent capture of the entirety of the mitochondrial genome, given it's almost the entirety of it, which is transcribed. Now, we could then use these mutations to attempt to perform lineage tracing, and we wanted to ask whether progenitors and their progeny might actually contain mutations in barcodes of these mitochondrial genotypes that might allow more closely related cells to be separated from more distantly related cells. To be able to test this in a toy situation, what we wanted to do is actually take initially a cell line, in this case TF1 cells, and iteratively derive a known experimental tree by performing single cell sorting over many generations over about 30 weeks. Using this approach, we can then obtain both mitochondrial genotypes using attack sequencing, as well as epigenomic information. As you can see here, the known experimental tree is shown on the left. And we can see different mitochondrial mutations that span either all subclones or specific subclonal populations, as I'm showing you here. By aggregating all of the information from all of the innumerable mitochondrial genotypes we obtained, we can actually get about 96% accuracy to reconstruct the known experimental tree without any a priori knowledge. If we then look within subclones, we're about 80% accurate to reconstruct the known lineage tree, really suggesting that even within more limited populations, if we can reconstruct lineage relationships or clonal relationships by using this approach. Importantly, we're not only getting information, of course, on mitochondrial genotype, but we're also getting information on variation in cell state using this approach. So we can start to examine how there might be variation in particular subclones, for instance, comparing accessible chromatin at one particular locus in these particular subclones colored in purple versus those colored in gray as shown here. Now, we then wanted to try to compare this to a gold standard using exogenous genetic markers, such as lentiviral barcoding approaches. And so we could barcode a limited number of these same TF1 cells, then examine them using the mitochondrial genotypes. When we do this, what we can see is the lentiviral barcodes are represented by these colors shown at the top of this graph. And we can see a variety of mitochondrial genotypes that actually you can see are uniquely distributed based upon the lentiviral barcode. If we aggregate all of the information that we have for mitochondrial genotypes and reconstruct the known lineage relationships, we can actually get about 95% accuracy to reconstruct the lineage relationships based upon these lentiviral barcodes. But in this case, we've not introduced any exogenous barcodes. We're simply using the endogenous mitochondrial genotype. So once again, demonstrating the value compared to gold standard barcoding approaches that are typically only only be taken advantage of in the setting of model organisms or using labeling in cell lines. Now, what I've shown you so far is really in cell lines. So we wanted to take advantage of this in primary human cells, particularly human hematopoietic cells. So we could derive a variety of myeloid and erythroid colonies from CD34 positive cells of a couple of donors, as I'm showing you here. And we could pick individual cells from each colony and subject them to single cell RNA-seq, in this case using SMART-seq2 based approaches, full-length single cell RNA-seq. And we could obtain about 1,000 or so cells that pass quality control. If we cluster the cells in TC space based upon gene expression alone, we see two clusters irrespective of donor. And not surprisingly, one of these clusters corresponds to the erythroid colonies or single cells from erythroid colonies that we had picked. And the other cluster is colored with hemoglobin. And the other cluster corresponds to myeloid cells from myeloid colonies that we had picked. And this is shown as colored with myeloperoxidase expression. However, in contrast, if we then use mitochondrial genotypes to cluster these colonies, we actually separate out the two donors from one another. And importantly, we can actually get unique mitochondrial barcodes that are found in single cells picked from the same colony, really suggesting that we can actually get more clonally and closely related cells and separate those out from more distantly related cells, even starting with the same progenitor population from the same donors, as I'm showing you here. Now, we've been able to use a variety of applications, but all of the technologies I've been able to show you until now have been limited because they really require us to use plate-based technologies, either single-cell RNA-seq with SMART-seq2 or single-cell attack sequencing using earlier fluidine-based methods where we can only profile hundreds or thousands of cells. So ideally what we would want to do is expand this to tens of thousands or hundreds of thousands of cells using droplet-based technologies. And this has been a major emphasis now for our group, is to try to actually expand these technologies and thereby expand the kinds of applications one can use these for. So what we could show was that commercially available approaches for single cell attack sequencing were beneficial, but both Bio-Rad and 10x Genomics in their commercial platforms took advantage of nuclear lysis. And this unfortunately got rid of most mitochondrial reads that I'm showing you here using, for example, the OmniAttack protocol from 10x Genomics. However, if we reverse engineer the approach that 10x Genomics had developed and simply perform a whole cell lysis using an NP40 containing buffer, we actually can get excellent capture of mitochondrial DNA fragments without any loss at all in epigenomic information from the same single cells that we were obtaining. Importantly, if we perform cell line mixing experiments, as shown here using these two human cell lines that are marked by distinct homoplasmic mitochondrial mutations and then subject them to single cell attack sequencing. If we do not fix the cells with formaldehyde, we can see that there's some mixing. But if we perform formaldehyde fixation with 1% formaldehyde, as I'm showing you here, prior to cell lysis, we get very little overlap between the cell lines. And in fact, we can then further discriminate these doublets by performing computational doublet discrimination. We've also further improved the process by actually being able to reconstruct regions that are homologous between the mitochondrial genome and the nuclear genome to enable excellent and robust mitochondrial genome reconstruction as shown here and get per single cell about 200x or so coverage on average, really enabling us to get excellent mitochondrial genotype calls down to a heteroplasmy of 1% or so. So then we wondered, could we take advantage of this to start to gain insights into human hematopoiesis and how this process goes awry in disease? And so at first we took advantage of in vitro situations where we could take a limited number of stem or progenitor cells as marked by CD34 and in vitro expand these and either differentiate these cells towards the myeloid or erythroid lineage. Now, when we do this, we could see that there is actually increasing accessibility either towards GATA motifs that occurs in an erythroid trajectory, or we could see that there's a trajectory of the stem or progenitor cells towards the myeloid trajectory as marked by CBP alpha and SPY1 motif accessibility, as I'm showing you here. This is by aggregating all the cells over the different time points that we've aggregated. But importantly, if we then look at mitochondrial heteroplasmy at the earlier time points and then compare them to what occurs as the cells undergo differentiation, we see actually two patterns. We see either enrichment of heteroplasmy or depletion of heteroplasmy. This is looking at one particular trajectory, either towards the myeloid or erythroid lineage. And importantly, we see that there's not even in balanced production as we would expect if the cells were equipotent, but rather there's biased production either to the myeloid lineage or towards the erythroid lineage, comparing day 8, where the cells have already undergone specification to the one lineage or another and undergo further differentiation. We can see this as exemplified here where there's, for example, bipotent clones where they're marking both the progenitor compartment as well as the myeloid or erythroid compartment, or we can see that there's biased progenitors as marked here in the erythroid compartment, the progenitor compartment, and later in the erythroid compartment, or as shown here in the myeloid restricted cells that are really restricted to this myeloid compartment. We could then use these different cells that are biased to different extents and quantify the degree of bias either towards the erythroid lineage or towards the myeloid lineage. And of course, I'll just remind you that this is all in vitro. And we could look at differential accessibility either for GATA motifs or KLF1 motifs in the erythroid bias clones, or more accessibility in the earlier progenitors in SPY1 and CBP alpha, even in the progenitor state that gives rise to these different cells. Now, the situation I've shown you is, of course, in vitro, and we wanted to start to look at situations where we could actually gain insights into in vivo hematopoiesis. And we started off by looking at malignancies, in part because these have limited clonality. So in collaboration with Kathy Wu, we've now looked at a number of chronic lymphocytic leukemia samples, in part because we can readily obtain a predominant malignant population that's oligoclonal in the peripheral blood. These are marked by CD19 positivity, which marks the malignant B cells. And we can subject these to a variety of single cell approaches. Now, by B cell receptor sequencing, we could see that the majority of individuals actually, or the majority of cells that we were profiling are actually these malignant B cell clones. But even within a single patient that has a single B-cell receptor rearranged malignant clone, there's actually some clonal populations that can be detected using mitochondrial barcodes, as shown here, for example, for a single patient, where each of the mitochondrial barcodes marks subclones of a single B-cell receptor rearrangement. But we can start to gain further insights by, for example, performing reference embeddings onto healthy peripheral blood mononuclear cells, showing myeloid cells, different T cell populations, NK cells, and B cells. We can see that the malignant cells have a variety of mitochondrial barcodes, but that across multiple patients, these are often shared across other populations, including myeloid cells, as well as other lymphoid populations, suggesting that even while CLL is traditionally thought of as a mature B-cell dyscrasia, there might be early stem and progenitor cell contributions that are resulting in this malignancy. However, our goal really has been to dissect normal human hematopoiesis using this technology. And I want to give you some examples where we've taken advantage of this approach in the last couple of minutes. So we can, for example, take advantage of surveying stem or progenitor cells from a single donor. And about 3 months later from that same donor, collect peripheral blood mononuclear cells, as I'm showing you here. We could, from each compartment, survey about 7,000 to 8,000 cells. By embedding the stem or progenitor cells from the bone marrow of this donor onto a healthy reference embedding, we can see that we get nice representation of stem or progenitor cells of different lineages, as well as excellent coverage of the peripheral blood mononuclear cells, as I'm showing you here across different compartments. Importantly, if we look at shared mitochondrial barcodes between the peripheral bloods 3 months later and the earlier stem and progenitor cells, we actually see excellent sharing with hundreds of mutations that are shared across these thousands of cells, really suggesting that we can get excellent coverage even after months of duration. Importantly, we can see that there are a number of clones, some of which are restricted to the stem or progenitor cell compartment, some of which are widespread. And this enables us to gain insights into the clonal distribution across different lineages. And I'll just show you one result here where if we look at the individual contribution of any progenitor to the mature peripheral blood compartment, each progenitor in this healthy individual is only contributing a minor amount, typically less than 1% of the clones within the peripheral blood. And as you can see here, looking across hundreds of different clones in the peripheral blood, they tend to have equal and balanced production of at least the detectable NK and T cells, monocytes, and B cells that are detectable in the peripheral blood. And so certainly this is an early result and we have much more to do, but I hope that this gives some insights into how we can take advantage of this technology of mitochondrial DNA barcoding, along with single cell genomic technologies, to be able to really dissect, hopefully, the hierarchies that give rise to normal hematopoiesis, and hopefully take advantage of this as well to better understand how this process goes awry in disease. And so with that, I just wanted to summarize by saying that mitochondrial DNA mutations can enable clone tracking and lineage tracing in human cells, that many single cell approaches can actually use and detect mitochondrial mutations. And I think as we're starting to develop higher throughput approaches and multi-omic approaches, we can certainly try to gain further insights into how this process might occur normally in physiology, as well as how pathologic aspects of hematopoiesis, as well as other processes, can occur. And with that, I just wanted to finish off by acknowledging the people involved in the work. The first vignette was really driven by Richard, Fulong, and Li Ming, Tao, and Aviv Regev's group, along with Blake Cohen in our group. And the second vignette I've talked about was really led by Leif Ludwig and Caleb LaRue in our group, along with another close collaboration with Aviv Regev's group. And with that, I want to thank you all for your attention. And I think that there's a few minutes remaining for questions, so I'm happy to take any questions. Thank you.
Great. Thank you, Vijay. Really great talk. I think I learned more technology that I was unaware of just by your presentation today. So thanks. I love the mitochondrial tracking and the DNA. That's actually a really smart approach. Anybody want to ask a question? They're welcome to just unmute themselves and ask at this time. Raman, do you want to go ahead and unmute yourself and ask your question?
Sure. So hi, I'm a graduate student at Yale and Andrew Shaw's lab, and I just want to thank you for a really interesting talk. It was thought-provoking, and I was wondering what extent can mitochondrial DNA clonality be called using the 10x single cell RNA-seq protocol, because we have a lot of those data sets, and it'd be interesting to go back to that and see if we can reanalyze it.
Yeah. No, thank you for your question. It's a great question. Unfortunately, the typical 10x genomics single cell RNA-seq workflows are usually 3' based. So they actually have very poor coverage of the mitochondrial genome. Certainly, you could occasionally get lucky and get, a mitochondrial mutation or two, but I think for calling it in an unbiased manner, it's not so helpful. So we have taken advantage of extreme situations. So for instance, if you have homoplasmic mutations and you do a stem cell transplant and you use data, for example, after transplantation, you can actually detect donor chimerism probably better than you can with conventional like HLA typing and things. So there's certainly applications, but unfortunately, it's a little bit limited in terms of the diversity. 5' based approaches using single cell RNA-seq have better approaches. Having said that, if you have a library, there are actually enrichment approaches, and Peter Van Galen has been actually developed at the Brigham and Women's Hospital, has actually been developing and has on bioRxiv a great method that he's developed to actually enrich for using 3' based libraries, mitochondrial transcripts. You don't get still perfect coverage, but you actually enrich for those using libraries that might already exist. You have to go back and resequence them, but I think it does give you a single cell resolution, great genotypes. In addition, the one thing I will say is in addition to Peter's work on enriching these RNA-seq libraries, the other possibility is with the release of the multiome kit from 10x Genomics, we actually now have seen that with some adaptation, the same method I've shown you for single cell ATAC-seq can actually work with the multiome kits as well with some adaptation. And so you can now get from a single cell, basically RNA-seq, ATAC-seq, and concomitantly lineage information using mitochondrial genotypes. And we've also combined this now with surface labeling. So basically you can do multimodal profiling. Some of this work will be published in the next couple of weeks in Nature Biotech, in a collaboration we've had with some investigators in New York Genome Center. So I certainly think that as we can do this, we can use these combinations. But the good part is hopefully we can adapt this across a range of technologies and really democratize it. So.
Thanks.
Any other questions? Just give it a minute. Okay. Otherwise, Vijay, thank you so much for your presentation. It was great. It was a really great talk, really interesting development of technologies that look like they'll be really useful for a broad population of the community studying hematopoiesis. And so thanks for sharing that. And thanks for those who've attended today, and hope to see you next month on July 7th, where Roland Lill from the University of Marburg will be presenting on the biogenesis of iron sulfur cluster proteins. And thanks again, Vijay, and everybody, please take care.
Thank you.